So thank you very much for inviting me today to talk about another receptor that we are investigating since, let's say, 20 years. We are involved in at the University of Perugia. It's not working. Okay, and use the keyboard. Okay, the nuclear receptors are some. It's a big family of receptors. That is, it contains mainly 48 receptors. Some of them have been deorphanized, so we know what they do in our body, in our cells, because we know the endogenous ligand. And one of them is farnesoid X receptor. They share a modular structural organization that is done by the N-terminal domain, a DNA binding domain, then an injury region, and the ligand binding domain. The ligand binding domain is the one that we try to hit by our compounds, and then they have also a C-terminal domain. The farnesoid X receptors can, as the other receptors of the nuclear receptors, can adopt a different conformation depending on the ligand that binds to it. And it also acts by modulating some homeostasis, like bile acid homeostasis or glucose homeostasis in the cells. So by changing its conformation. It has also, it can work as homodimer or heterodimer with FXR, that is another, that is the retinoid X receptor. But it's not, it can work in both the situation, as monomer or heterodimer. So we have different conformation, not of FXR, because we have some apo X-ray structure, so without the ligand inside, but the conformation I can say is not too much different with respect to the ligand binding, the one that binds the ligand. So the allo is not so different in the case of the apo form in the X-ray of FXR. This is not well understand, because in other cases of nuclear receptor we can see this shift of this helix, that is a functional helix that is change its conformation depending on the ligand that binds to the binding site, and depending on the function of the receptor. So it can bind a co-repressor or a co-activator, that is a peptide. Then it binds the other partner. Then it goes to the transcription of the genes, giving the overexpression of some proteins to have a regulation of this equilibrium of lipids and glucose in our cells. So in our case, we will talk about FXR. In my case, I will talk about FXR. FXR stands for farnesoid X receptor. Farnesol was the one, the endogenous ligand that was thought to bind to FXR, but later it has been understood that the real binder is the canondesoxycholic acid. So it's a bile acid. So it mainly binds bile acids, and it regulates the bile acid homeostasis in our body. FXR so binds CDCA. The binding reflects on the changing of its conformation, the coactivator recruitment, the FXR dimerization. So it goes on the transcription of some genes that are involved in the bile acid homeostasis. What happens is shown in this animation that is not a molecular dynamics. I want to underline, it's something just to show you what happens. So when kinetotoxicity binds to FXR, it starts to lose the coactivator in cyan. Then the AF2, that is the H12 helix, binds to the receptor. Then it recruits the coactivator, and then this goes to the transcriptions of some genes. And so you have the modulation of the expression of these proteins. In our efforts in Perugia in 2002, we, in the Professor Perugia group, developed the obeticholic acid. So by the insertion of this 6-alpha ethyl in this position, we passed from the 10 micromolar potency to 100 times more potent compound. So it's been a great boost. And this was in collaboration with the Intercept Pharmaceutical that lead to the FDA-approved drug for the primary biliary cholangitis in 2016. So we were quite interested in this receptor because we were first in class. We were in collaboration with the company, so they wanted to explore also different bile acid substitution to cover the patent, of course, but also to understand better the FXR receptor. And this is the reason why this happened. In 2002, the kinetotoxicolic acid was also in X-ray structure resolved with FXR. So we understand that there was a hydrophobic cavity that was free to be filled up by an ethyl group. So this was the magic 6-alpha ethyl substitution in our case to have this improvement in potency. In our focus, we were always interested in understanding better this receptor. So this is why we were interested also in some antagonists. They were thought to be antagonists, like gugusteron. Gugusteron was it's a natural compound. It stays in ENZ disposition, and so it binds to FXR, but it doesn't give any effect in the transcription of the proteins. But in contrast, it can antagonize the effect of the agonist. So, in order to understand better this, we studied a little bit, and we have seen that gugusteron is not a clean compound because it binds to more than one nuclear receptor. So what you see in the cell as an effect of cholesterol reducing effect can be attributed to different interaction that the gugusteron has in the cell. By the way, we performed some assays, and using the gugusteron together with the INT-747, that is the obeticholic acid, so the compound we developed, we have seen that it was possible to see a reduction in the effect of the agonist. So in this line, we started to do some in silico analysis. So we started with the docking. We did it was in 2005 by Udo Meyer that applied in that case ALLODD. So by computational docking with a quite big grid, he has noted the possibility to host the gugusteron molecule in a non-canonical binding site, so not where the CDCA was co-crystallized, but in another site that was never been disclosed before. And this was just an observation that we proposed as an hypothesis of binding. And so we tried also to investigate deeper this possibility. Here you see why the glucosterone can bind here. Glucosterone is quite not so big molecule, so it can be host here. And it can be hosted together also with the chenodesocyclic acid or other agonists that they don't interfere each other. And it's quite hydrophobic, this site S2, so it can bind to glucosterone. We tried also to investigate if it was possible to reach the S2 site by elongating the side chain of the bilasin by some substituents that were quite hydrophobic, as you see, and also not too much flexible. So we were quite confident also in the docking proposal, because in the meantime also the obeticholic acid X-ray was resolved. So it was mainly occupying the same disposition of the CDCA. So it was perfectly fitting the hypothesis of the binding potency increasing by the ethyl occupying the hydrophobic interaction before. And we have seen that in this case, it was possible to generate a partial agonist by inserting this substitution on the side chain, but you have a partial agonist if you remove the 6-alpha. So with respect of the obeticholic acid, you have to elongate the side chain to have a partial agonist. So partial agonist means that when you have the transcription of the proteins, you don't have the full effect that you see in the full agonist. You have a sort of a middle effect with respect of CDCA and an half effect with respect of the CDCA transcription. And but you have to have a partial agonist, you have to remove the 6-alpha. So you can see here the efficacy that is 15% with respect of the full agonist that goes up to 100%, because it's referred to the CDCA, that is the natural binder. So after a few years, in 2014, these guys made some deuterium exchange experiment on FXR, and they found nicely that GW was a Glaxo full agonist developed the compound, because after obeticholic acid, a lot of companies started to work on FXR. Eli Lilly, Roche, Pfizer, Glaxo, and so on. This is one of the reference compound, we can say. So here by the deuterium exchange, you see that the disposition and the part that are touched by the binding of the GW with respect of the apo form of FXR. With the CDCA, the violet part is this. And with the glucosterone, you see that there is a different part with respect of the full agonist, because you have the canonical site, but also a part here in the upper that is never violet in the other cases, that corresponds, that is in agreement with the hypothesis of binding of glucosterone to the S2 site. So they found mainly that glucosterone was able to bind the canonical binding site S1, the CDCA1, and the S2 site. So it was possible to bind both. We performed also some antagonism profiling in vitro, and we understand that the profile of the curves were indicating a non-competitive antagonism effect. So non-competitive means that they bind in different places, so they are not competing for the same site. but you have a reduction of the effect by non-competitive antagonists. So the full agonist binds on the canonical site, the glucosterone binds on the S2 site, and then you have the reduction of the effect. So this was further confirming the S2 site binding hypothesis. And we also tested this hypothesis with these compounds, especially with the one that was full agonist occupying the same S2 cavity. And we observed what? We observed that there was a reduction. So in the case of obeticholic acid, you see the reduction. In the case of UPF 930, you see the reduction. What does it mean? We investigated this biomolecular dynamics and endocrine simulations, and we have seen that these carbamates, modified derivatives of obeticholic acid, were able to bind both on S2 disposition, so occupying the same of the one of the glucosterone, but also they were able to shift this arm on another S3, we call the S3 site, on another site, we can say, that is more solvently exposed, that can host still the R, the side chain R. This is compatible with what we have seen here. So this reduction can be attributed to the fact that the full agonist, 930, changed its conformation upon the glucosterone binding on the S2 side by shifting the arm on the S3 disposition. And this was compatible with our in silico studies. This is also another paper that we published quite recently, in 2019, describing this. But we were also focused in understanding this better, and we found this paper by Daniel Mack. Daniel Mack is another of the key player in the FXR targeting. And in 2020, he published this paper where he found that these imatinib derivatives called P16 was able to give an antagonist effect or an overactivating effect. So it's an allosteric modulation. So what is nice is that they found a non-competitive still compound able to potentiate, in this case, the effect of a full agonist. Few years later, another group used this data to perform some computational studies. And what they found is that the P16 was compatible in the binding within the S2 side. So further confirming the possibility to occupy this allosteric site by glucosterone to have an antagonism effect or a potentiating effect given by this P16 compound. And if you see here, we made a superposition of the glucosterone side and this P16 disposition, and they are quite overlapping. So, in conclusion, I still want to understand, but there's a, because everything started from ALLODD. So ALLODD was the only program in 2002 able to say, oh, glucosterone could bind here. We tried to reproduce also this data by other softwares like GOLD, by Cambridge Crystallographic Database, also by Glide of Schrödinger, Swift. But we failed always. You have to force this program to say bind the glucosterone. So it's hard to understand. So ALLODD was really good in giving this hypothesis at the time. So I wanted to change this paradigm using another docking programs that now are on the high, you know, because they are based on artificial intelligence. And I'm talking about deep dock, neural plexer, and dynamic bind. So I've tested all these three softwares to understand if they were able to bind the boost around to the S2 side by unbiased, without any me to force them to bind that. And because they approach the docking in a different way with respect to the normal programs. They start to they use the diffusion approach, and they start to describe the ligands. Then some of them reconstruct the protein outside the ligand and gives you some hypotheses. So it's a different approach with respect of creating a grid inside the frozen protein and so on and so on. I was thinking they could be successful, but it's not the case. All of them failed, still failed. No, they are not working. And the reason I could say this is, I'm approaching the end of my talk. The reason could be the fact that the allostery is not so frequent. It's not so frequent also in the X-ray structures. So you don't have this information even in the X-ray structures. So you don't have such information to be used by the artificial intelligence models, because they don't have this information. Simple. And the other stuff is that they describe the ligand, always seeing what they find in the PDB-bind database. So you have still normal, let's say, or canonical binding site discovered and described. This is the reason why we are still failing. So the allostery, in our case, in this case, is still a complex, a real complex problem to solve. And everything started in our case by an observation in which we trusted at the time. This indication was coming just by one software, and we didn't, we followed this indication at the time, and we have confirmation during the years by our compounds, by our group, but also by other groups and our research papers. And with this, I will conclude by thanking all the guys I'm working on with Perugia, and also Professor Chen and Pharma for their collaboration, and Institutet Pharmaceutical for the support of the research. And thank you all for your kind attention.
We have time for one question. We have one of our slides. Yeah.
Thank you for the talk. So two questions. First, like, how do you define that that's two pockets? I understand through ALLODD. That's kind of a weak, maybe point to start for the developing of a drug. So did you assess the credibility of that pocket? How did you fall to go through with that one? You just using the ALLODD, or, I mean, I suppose you did some other analysis. And the other question, I mean, compared especially to other maybe pockets as 3 and so on. And the other question is, I saw that your developed molecule, you also designed the linker, let's say, to connect that part that is able to bind to S2. How did you design the linker? I mean, of course, I don't want to waste all day, but just maybe you can say a little bit more about it.
Yeah, sure. So coming the first question, this was just a starting indication. So we have to say, I have to say also that we were interested in FXR and in full arm. Okay, but you know, when you are focused on a receptor, you want to know almost everything, how it behaves in different situations with different counterparts like ligands and so on. So the allosteric was something really nice to understand, especially because FXR in full agonist could go in overactivation, giving some side effects like pruritus and so on. So also the companies were trying to find something to modulate a little bit this overactivation with respect of this DCA. So we were interested in this aspect. We started with this indication on the ALLODD, but yes, later we did this experiment that I show you by antagonists, and we try to understand if they were competitive or non-competitive, and you can see it by the profile of the curve. So you have indication about supporting this hypothesis, of course. Later, we did also mutagenesis studies. I didn't show because of time restriction. And some of the mutagenesis confirmed, but some of others didn't confirm because we touched probably some particularly important amino acid signaling the effect coming from S2 toward the S1 and then to the F2 helix and the coactivator recruitment. So it was quite hard, but some pieces of the puzzle were composing the whole picture, and it was quite nice, but it took a lot of time. Going to your second question, we did this type of exploration because we had the X-ray. So by docking, you design something, you see which type of chemical synthesis you can perform without, because the biological chemical synthesis is quite hard. It's a quite hard task. You're not allowed to do whatever you want. But we explored this possibility, and we decided to go with the carbamates because they were quite easy to do and quite stable. So for these two reasons, we reached the S2 site, and we found good results. Now we are writing a paper on partial agonists of FXR, going through different biacid position derivatives. They are not so far, they are not going to AS2, but they are going to the canonical binding site, the part that is more in touch with the helix 12. So it's still something that is going on, and the exploration was made due to the fact of not giving also the possibility to the companies, other companies, to break the patent and find some holes in the patent with some good compounds. So there were two. One is the research point of view, the other was to cover also the patent and for the company.
Thank you.
Thank you very much for this beautiful presentation of your very successful drug design that you presented to us. I have a comment about the interpretation of the data. We have been ourselves working on some transcription factors, and there has been also a very expanded study with the LAC repressor that you may know, where all the express structure has been identified, and where both the extras, the globular binding site and the DNA binding site have been analyzed separately. And it's clear that if you want to bind to a DNA molecule, you need to have two arms. So it means that the active conformation in vivo of the receptor is an actual dimer, and strictly dimeric. And what we found and published 2 years ago for some of these transcription factors is if you have some compound which dissociates the dimer, then they act as very powerful competitive antagonists because the monomer can no longer act on the DNA and be effective. So most of your studies are on the globular domain. Have you been doing the X-ray structure of the whole protein with the DNA binding domain or not? And my question is whether some of your data could be explained by the actual interaction of the two domains together and see whether some sites are at the boundary of the subunit or not.
Yeah, we are trying to investigate also this aspect, of course, because unfortunately there's no complete structure of FXR together with also RXR, but it's present in LXR, that is a different but quite near protein with FXR. So we are trying to make some simulation with this, but also in literature they are saying that it works as dimer. Some of them, just a couple of papers says that it works as homodimer, but it's quite strange. And but we don't have the full the first structure. But we know the problem that when you are seeing the small part of a bigger picture when you are when you are working with the ligand with the nuclear receptors. And
okay, let's take from Andrea again.